Hello, welcome to this video on DNA. This is the first video on DNA. This is the one on nucleotides. So let's define what we're talking about here. When we're talking about DNA, this stands for deoxyribonucleic acid. That's where the DNA comes from, and that's a polymer that contains all of the genetic information of an organism. It's a condensation polymer, all right? And nucleotides are the monomers. The nucleotides are the monomers, and on the right-hand side here in this picture, we've got a schematic of a nucleotide, and it has three groups. We have a phosphate group, a pentose sugar, pent as in 5, and you can see 5 points to the pentagon there, and then we have a base, of which there are 4. So this video is on the nucleotides and how they're structured and how they come together from the different groups. So let's have a look in detail at nucleotides. So we'll bring up the structure that we had on the previous slide. Here it is, where the nucleotide is made up of 3 components: the phosphate, the sugar, and the base. And we're going to look at each of those components in turn. Let's start with the phosphate. So the phosphate is an ion, an anion, a negative ion. And let's have a look at that over here. So we've got a phosphorus atom with double bond oxygen, and then single bond oxygen here. 2 of those oxygens are bonded to hydrogens and with hydroxyl groups, and the hydrogen that was on the remaining oxygen has gone. However, that oxygen has kept its electrons, so it's got a negative charge there. So that is the phosphate ion that occurs in nucleotides. Sometimes all 3 hydrogens are lost, as in phosphoric acid, where you get a PO4 3- ion, but in the phosphate ion that occurs in nucleotides, we retain 2 of those hydrogens, such that the phosphate ion only has 1 minus charge. Okay, so that's the phosphate ion, and that's the same in each nucleotide. Let's now have a look at the sugar, and this is a pentose sugar. It's a pentose sugar. You can see the pentagon shape there. And there's 5 carbons. Although when we draw it out in structural format, there's an oxygen atom at the top of the pentagon. That's bonded to a carbon atom, carbon atom, carbon atom, carbon atom, back to the oxygen. So each of those corners, as you know, is a carbon atom, and the carbon atoms are in turn bonded to other groups that I'm going to draw now. So let's first number the carbons. Normally we number the carbons, with this being number 1, 2, 3, and 4. So starting on carbon number 1, we have a hydroxyl group and a hydrogen. Carbon number 2 just has 2 hydrogens on it. Carbon number 3 has a hydrogen and a hydroxyl group on it as well. And carbon number 4 actually has a different sort of group. It has a carbon that's bonded to 2 hydrogens and a hydroxyl group, like so. And this particular sugar is called 2-deoxyribose. 2-deoxy, not dioxy, deoxyribose. It's a derivative of the sugar ribose. A straight ribose would have a hydroxyl there on carbon number 2, but in 2-deoxyribose, you've lost that oxygen and you've just got lost that whole hydroxyl group there. That would be, the red would be for ribose. All right, so we instead have a hydrogen in 2-deoxyribose. So the 2 comes from it being on carbon number 2, and the deoxy means that you've removed the hydroxyl group. So deoxyribose is the sugar, and that's the same in each nucleotide. So moving on now to the bases. whereas the phosphate and the sugar are the same in each nucleotide. We have 4 different bases. The first of which is called adenine, and that looks like this. And the nitrogen that we have circled in red there is the nitrogen that bonds to this carbon number 1, when that forms the nucleotide. Okay, so adenine is one of our bases. Another of our bases is called guanine. Again, we've got the nitrogen that makes the bond circled in red there. The third one is called a cytosine. And again, you can see the nitrogen that makes the bond. And the fourth one is thymine. And again, you can see the nitrogen bond there. So we can make 4 different nucleotides. And that's what we're going to have a look at next. So we're going to draw out the structure of these 4 nucleotides, and they of course come from the 4 bases. So let's just remind ourselves of the name of the 4 bases. We've got adenine, and you do well to memorize these 4 bases. We've got guanine, adenine, guanine. We've got cytosine, and the fourth one is thymine. So they're the 4 bases, and they make 4 different nucleotides. But as you remember from the last slide, then the sugar and the phosphate are the same in each case. So here we've got the phosphate and the sugar joined together, and you can see that this carbon here remains from the original sugar molecule, but the rest of the molecule has gone. The H2OH has gone, and the phosphate has joined with it. But the purpose of this is to draw the nucleotide, which entails bonding our base onto our sugar phosphate complex. And if we're going to draw adenine nucleotide, right, we need to draw the adenine onto this. So adenine looks like this. We're bonding onto carbon number 1, and we have the pentagon hexagon structure, which we're going to draw out like this. And that's a double bond. And that's also a double bond. And the top one is a hexagonal shape, like so, with an amino group coming off this carbon here. This is a nitrogen, and this is a nitrogen, and we've got double bonds here and here. You get the structure of these in the data booklet, so you don't need to memorize these, but that is the adenine nucleotide. So let's label that one. That's the adenine nucleotide. And in the exam, you need to be able to do what I've just done there, draw the structure of it, but you are given the components in the data booklet. Okay, let's draw the next one. Again, we've got the phosphate sugar complex already drawn for us, so we just have to add on the base. Now I'd like to add the base thymine to this. So thymine is going to, let's draw it in different color to the light blue that we used for adenine. So this one we're going to use red for, and we're drawing thymine now. So again, we've got nitrogen making that bond there, but we've just got one hexagonal structure here in thymine. This carbon is double bonded to an oxygen, and let's complete our hexagon shape. And this corner is actually a nitrogen hydrogen, right? This carbon is double bonded to an oxygen, and we've got a methyl group coming off this hydrogen here, like so, with that double bond there. So that is thymine nucleotide. So let's label that. That's thymine nucleotide, and I've drawn those in that order for a reason. I'll come back to the reason, but they pair up when forming DNA. They pair up. So let's have a look at the other pair of nucleotides. Again, we've got our structure to start off with, and we're going to draw guanine. So guanine is also a pentagon-hexagon structure, but as usual, we've got a nitrogen bond here, and the pentagon is drawn first, like so, and this opposite nitrogen is also in the structure with a double bond, and then attached to one bond of the pentagon is a hexagon, like so, where this carbon here is double bonded to an oxygen. This is actually a nitrogen-hydrogen group, and this carbon is bonded to an amino group, and we have a double bond to another nitrogen here, and this joining bond is a double bond as well. So that is our guanine nucleotide. Guanine nucleotide, and that pairs with cytosine. All right, so let's draw our cytosine in. We can use purple, I suppose, for cytosine. Like before, we've got a nitrogen that makes the bond, but also like thymine, we've just got a hexagonal structure. Here's our hexagon, and this carbon here is double bonded to an oxygen. This is actually a nitrogen on that corner, and it's double bonded to this carbon that is bonded to an amino group. And lastly, we just complete it by drawing our double bond in there, and that is the cytosine molecule that is makes up the cytosine nucleotide. Cytosine nucleotide, and they become the four nucleotides, and let's just remind ourselves that they pair up. Okay, so let's pair them up. So thymine pairs up with adenine, and guanine pairs up with cytosine nucleotides. Okay, and in the next video, when we look at DNA, we're going to look at the pairing in more detail that involves the hydrogen bonds. Okay, but we're going to leave it like that for just now, because first of all we have to work out how they form polynucleotides. So at the moment they're single molecules, 4 different single molecules that are monomers, but they take part in condensation polymerization reactions, all right, to make polynucleotides, and that's the subject of the next slide. Okay, here we are looking at polynucleotides and how they're formed in this condensation polymerization reaction. So let's start off with one monomer, and you can see we've got a phosphate group at the top. We've got the sugar group, as you can see the pentagon there, and we've got a generic base. It could be any of the 4 bases. It doesn't matter for these purposes which one it is. Okay, so that forms our monomer. That is the nucleotide, and we're going to make one link, all right, to make a dimer, for which we need another monomer. All right, so we've got another monomer. The base could be the same, could be different, but the important thing here that we're learning right now is how we make the link between 2 monomers. It's a condensation reaction, which means, of course, that you get a covalent bond between the 2 monomers, and in the process of making that covalent bond, you drop out a small molecule. That's the definition of condensation reaction. And so the hydroxyl group from the phosphate, that's this group here, reacts with the hydrogen bond of the hydroxyl group coming off the top sugar. And let's draw a circle around those because they are the ones that leave. All right, so they leave to make a water molecule. That's the small molecule that drops out. And then the remaining oxygen that I just draw an arrow to makes the covalent bond with the phosphorus from the other nucleotide. So that results in the following molecule being made. Okay, so we start up here. We can draw our hydroxyl group from the top. We're just copying out just now. That's our phosphorus, double bond oxygen, right? And this is all the same as in the top. That's our oxygen that is bonded to our base, and our base we draw as a pentagon. Bonded to the sugar, the base group bonded to the sugar. And now this is the new bit. We have the oxygen atom, which is this oxygen atom here, bonded to a phosphorus atom here, which is this phosphorus atom there. The rest of the molecule is as on the left. Carbon there at that corner, and bonded to the bit that was the sugar. We've got our base, and we've got the remaining hydroxyl group hanging off the bottom of our sugar there. Okay, so what remains to be said is that if we go up to the top of this dimer and look at this hydroxyl group that's on the phosphate, well, that can bond with a sugar group of another nucleotide. So it can bond with sugar group, just as we've just done. It can bond with a sugar group of another nucleotide. And if it did that, you would have three nucleotide molecules joined together. And in the same way, if we go down to the bottom, this hydrogen here, that could bond to the phosphate group of another nucleotide. So could bond to the phosphate group, again, just as we've done on the left. Phosphate group of another nucleotide, like so. And you can see how the polymer molecule could build up, and each time that bond, another nucleotide is added to the chain, you get another water molecule dropping out. And if you carry on and on, you get a polymer molecule that can be represented like this diagram here on the right hand side there, where we've got a sugar molecule, sugar part of the molecule. That's the sugar part. This is the phosphate part. And then of course we've got this part is the base, and you can see that you've joined 1, 2, 3, 4, 5 nucleotides together in this arrangement, and it all looks quite tidy. So you've got a polynucleotide here, and DNA actually consists of 2 polynucleotide molecules that hydrogen bond together, and that becomes the subject of the next video. Just one last thing to do before we finish, and that is to introduce possibly a new word phrase to you. So when we created the link between the 2 nucleotides, we created what's called a phosphodiester bond. And so I'm going to circle around it here. So it includes the oxygen that came from one nucleotide, and it includes the phosphate part of the other nucleotide, and that, when together, makes a phosphodiester bond. So let's write it out. Phospho, from the phosphate part of the ester, diester bond. In a different topic, you've seen a similar thing called a peptide link, but in this case, our oxygen is also providing the link, but it's providing the link between a sugar on one nucleotide and a phosphate on the other nucleotide, and so it's called a phosphodiester bond.